On behalf of all the employees at ViaCyte, it's a pleasure to give you an update on the progress we've made here at ViaCyte. Our strategy really evolves around three things. We want to take and leverage our proprietary and deep expertise in stem cells and then combine that with innovative approaches in device delivery and immune evasion. And in device and delivery and immune evasion, we have bolted on partnerships to help enhance our efforts there. For example, we are partnered with CRISPR Therapeutics to develop a gene-edited cell line that would disintermediate the need for immunosuppression. And we partner with W.L. Gore, a worldwide leader in biopharmaceutical materials, to help advance and engineer our devices. We enjoy great partnerships with our investor syndicate, led by Bain, RA, and TPG. And we recently raised $45 million of money in a D round with Adage Asymmetry and Invis. These are exciting times for us at ViaCyte. And our initial focus is going to be on type 1 diabetes, and we've had significant milestones that we've delivered. This summer at the ADA, we revealed data on a patient that showed glucose-responsive C-peptide, a marker of insulin, that also maintained glycemic parameters, a significant development in humans for the industry. We're also going to be putting into patients by the end of this year a gene-edited cell line, testing for immune evasion and safety with a bridge to an efficacy trial in 2022. This would not require immune suppression. Third, we're going to be working in the portal vein, which is a device-free approach, similar to a cadaver islet cell transplant, but with ViaCyte cells, initially with the immune suppression, but eventually with immune-edited cells. And then finally, once we have this rolling, we're going to be looking in parallel to see what other diseases can we attack and solve. Why do we choose diabetes as our first target? Well, certainly there's a lot of unmet need. We've heard several speakers talk about that already. Type 1 diabetics do not produce beta cells, and as a result, they do not produce endogenous insulin that can be measured and marked by the lack of C-peptide, which is a biomarker for endogenous insulin. Then what patients do is they take exogenous insulin, which is certainly life-saving, but not completely optimal for the patient in the management of their disease. There is a high burden of disease management and a high degree of economic and morbidity costs. Here you see 8.4% morbidity in patients with just hypoglycemia alone, and almost $15 billion of cost associated with the management and cost of managing hypoglycemia. Our strategy is to provide a physiological artificial pancreas, implement a device with cells that can produce both glucagon and insulin and keep people in a counter-regulatory and regulatory management very close to normal. The great thing about it is we're far advanced to where this is no longer a dream, but this has got real potential. As you can see, our three platforms that we're looking at here, our lead program is this PEC Direct program. This leverages an open device. That is a device that allows direct vascular access. It uses our proprietary stem cells and does require immune suppression. This is the data that I'll show you in just a minute. Therefore, the patient type here that we're targeting is the high-risk patient, the hypoglycemic unaware patient, patients that might already be on immunosuppression. The goal, though, is to treat the entire population without the need of immunosuppression. That's where gene-edited comes into play. Our work with CRISPR Therapeutics has advanced several cell lines, and we're going to be putting those cell lines into patients, as I mentioned, and that will be testing for immune evasion and safety, bridging to an efficacy trial in 2022. Further, we're working with James Shapiro at the University of Alberta, who is a worldwide expert in cadaver transplants, to use a device-free approach using the portal vein. That will be initially done as we stand in a preclinical posture here with our PEC cells, but eventually with the gene-edited cells, we can put those directly into the patient. This is a little bit of the data that we displayed or talked about at the ADA this summer. Here you see a serial mixed meal tolerance test from a baseline week 26 and week 39. This patient had no detectable C-peptide and had the device implanted. As you can see at baseline, under laboratory conditions, showed no C-peptide, and glucose, as you would expect, continued to rise. By week 26, the ViaCyte cells start to kick in, and certainly by week 39, now you're starting to see a very impressive physiological response from the cells in our device. There is a C-peptide response of 0.8, and as you can see towards the end, both glucose and C-peptide are regulating themselves. A very, very impressive result. Getting C-peptide or insulin and glucose under control is one thing, but obviously you have to see what happens to the patient in terms of their glycemic parameters. I can report that the A1C that was dropped was sustained, but most importantly, as you can see from this picture, big improvements in the patient's glycemic control. This is a 2-week continuous ambulatory glucose monitoring graph. The dark lines represent the 70 and 180 milligram per deciliter glucose control levels that the ADA has determined as time in range. Pre-implant, this patient was afraid of going to be hypoglycemic. Therefore, she was 45% of the time outside the range and high. She was on 20 units of insulin a day, and she was in range only 55% of the time. Our last measurement of the patient, the most recent 52 to 54 week, you can see the patient now is in time and range 94% of the time. And I can report this patient is on less insulin than when they started the trial. Certainly our goal would be to be insulin-free, and we will be working to optimize our therapy to make sure that happens. But this is a spectacular result in a human patient, in a type 1 diabetic patient, showing that a 94% time in range, less insulin than when they came in, and of course, better opportunities to avoid major macro and microvascular complications because they're in range 94% of the time. What is significant about this result, in addition, is that we have actually de-risked the program when we look forward at our gene-edited line. The same cells and the same device that just produced the result you saw in a patient under immune suppression is going to be the same program that we're going to be using with our CRISPR cells. The edited cells that we have are designed to evade the immune system. So the only thing changing in this experiment is the efficacy of the gene edits. We have 2 knock-in, 2 knockouts, and multiple lines after that. Eventually, we're going to be seeing if these cells can avoid the immune system in a device and not have to have the need for immune suppression. Again, then we would move forward to an efficacy trial. This is an exciting development because it brings into all patients who are on insulin, including type 2 and type 1 patients, without the need of immune suppression, and it provides a reliable, tested source of cells as opposed to transplant patients waiting for donors. Speaking of donors, one of the big opportunities is how to disintermediate the device completely. We've partnered up with James Shapiro, who, as many of you might know, the architect of the Edmonton Protocol at the University of Alberta. He's a strategic advisor, and we're working with him to advance our program under his supervision to use our cells initially in a preclinical model testing for safety and feasibility. But in parallel, we're waiting for the gene-edited cells to kind of get tested and to be proof of principle. Those two things will converge, the techniques we're developing with Dr. Shapiro and the regulatory testing of the cell lines with gene-edited to eventually put the gene-edited cells directly into patients. So this would be a really, really exciting opportunity, again, for patients without the need of immunosuppression or a device. We have a couple of other things that make, I think, ViaCyte a very compelling opportunity. First, I would say our freedom to operate is very robust. We have over 850 patents that go out to end of 2037. They incorporate what the cell becomes, how you differentiate them, what the cell produces, and even the combination of them therein within the device. So our IP is very robust. And second, we've made significant and conscious decisions to invest in people, material, and processes on manufacturing. Today, we have a 6,000-foot clean room. Tomorrow, we're going to have 2 clean rooms of 12,000. We'll be doubling our square footage. We're scaling up to bioreactors, and we've made thousands of devices under GMP conditions. I would say that about 50% of our organization today is in some way, shape, or form involved in the manufacturing and release testing of our products. So we feel really good about our IP. We feel really good about our ability to make the products. We feel really good about the clinical progress we've made, and we have great optimism about the future. You know, in summary, our opportunity is to treat major unmet needs and go for a functional cure or a significant altering of a patient's lives. We have a significant lead. We are not in rodents. We are not in rats. We're not in models. We're in humans. We're also going in with gene-edited cells. We're well capitalized. We have good IP, and I think we have a bright future. So I think that the opportunities for us in the near term are significant. They're going to keep us on our toes, but they're exciting, and I think I share the same thing all of you do as well, which is patients are waiting. So with that, I will say thank you, and I don't know if there are any questions.